All right, time for part two of Module 6. Let's talk about brains. Um, this module is called Psychology Towards the Turn of the Century. So I guess that vaguely means from 1975 to 2000 ish. And it wouldn't be too inaccurate to say that the brain really started to take center stage in the field of psychology. It wasn't completely sidelined before that. Obviously, physiological psychology was always a very active uh, area in psychology. If you go read or flip through any psychology textbook from the late 1800s to the early 1900s, there's always a good bit of talk about physiology. But in the last quarter of the 20th century, we developed the methods to really finally start to look at the human brain in action. Because before that, we were looking at neurons uh, and petri dishes, and we were looking at animals. You have animals learn something, and then you do histological studies. You do slices of the animal's brain and put it under a microscope and do different dyes. There was kind of a gulf between human behavior and human mental processes and the very basic stuff we were able to learn about the brain before that. So, one of these technologies was the PET scan, positron emission tomography. It measures the breakdown of radioactive activity within the body. So, you're required to be exposed to a little bit of radioactive material, which poses some health risks. MRI without the F was invented alongside the PET technology around the 1980s. So PET stands for positron emission tomography. MRI stands for magnetic resonance imaging. And the PET scan was the better technology to use, they thought at first, especially compared to MRI. So in 1988, the first application of PET scanning was done to study cognitive function, and it was published in some of the top journals, Science and Nature. If you ever publish in one of these journals, then you are, I was about to say a made man, but I guess you could be a made lady, made man, made non binary, whatever. There are problems with PET scans. It's better than nothing if you want to observe the human brain in action. However, the real game changer came in the early 90s when someone modified the settings, the programming of an MRI machine to do functional magnetic resonance imaging instead of merely magnetic resonance imaging. And in 1991, the first fMRI was used to measure a difference in brain activity. So, what an MRI is doing, see, a PET is looking at how some radioactive material is broken down and cycles through your body. The MRI uses a different technology where it takes advantage of the different tissues, the different molecules, and the um, different tissues like bone, marrow, different neural layers, white matter versus gray matter, have different concentrations of different materials and different chemicals, and those chemicals and different concentrations respond to electromagnetic waves in subtly different ways. So if you get a very big, powerful magnet and spin it around a person, you can develop images that are inferred from the different ways that these different molecules and different concentrations respond to the electromagnetic stimulation. I'm not terribly concerned that you know a detailed story of how exactly the image results, but just know that what you're really looking at with MRI is a response to electromagnetic properties to tissues. With functional fMRI or functional MRI or fMRI, What they're doing is they're taking advantage of the fact that when you use one part of your brain more rigorously or more regularly, it needs to be replenished with, blood oxygen levels need to be flooded in so that the neurons can continue firing. So, slightly like a second or two after a neuronal region needs it, a blood oxygen level difference, or BOLD, actually stands for blood oxygen level dependent. The BOLD signal is a signal that more blood oxygen was needed in this area. It's being replenished in this area. That means that area was being used. These all came out in the late 80s and early 90s. And fMRI and functional MRI really, like I said, took off. In 1992, there were only a few publications that mentioned fMRI or functional MRI or functional magnetic resonance imaging. Over the years, though, they started picking up. Let's see, around the year 1999-ish, there were 500 publications that talked about MRI, and the trend speaks for itself. It keeps growing and growing in popularity. fMRI is where it's at if you want to see the living human brain in action. So let's talk a little bit about the properties of these different neuroimaging technologies. One of the most important things to consider if you're going to use a neuroimaging technology is its temporal resolution, that has to do with time, temporal, versus its spatial resolution, which has to do with space. If you have high temporal resolution, that's like a camera being able to take a picture of a speeding car and capture it from one frame to the next. You have this clean image of the car moving along its path. If you're using a camera with low temporal resolution, then you have motion blur. So you're capturing that something happened in that very general time frame, but you're not getting an accurate precise image of what happened for thin slices of time. You just kind of get this big chunk of time. There was stuff going on. So temporal resolution matters. And then you have spatial resolution. Spatial resolution has to do with picking up on fine details in space. So this image at the top left right here represents high spatial resolution. You can see the details of the car. With low spatial resolution, it's blurrier. You have less of a detailed look of the image itself. Best of both worlds is if you could have high temporal resolution and high spatial resolution. That's where you can capture things happening in real time very quickly, and to touch on the spatial resolution, you get accurate pictures within those thin slices of time. Worst of both worlds is low temporal resolution and low spatial resolution, which means you're not getting a very fine-grained image, or it's all kind of blurred together time-wise, and space-wise, you're not getting a very sharp image in each time slice. It's all very blurry. So I'm going to talk about the pros and cons somewhere down the line, but let me just say that fMRI really excels at spatial resolution compared to other neuroimaging technologies. I'm going to talk a little bit more about this later, but an fMRI captures pretty darn good how to put it. If you think of like pixels in a JPEG, it's a very high definition picture. fMRI can pick up on little teeny tiny differences in neuronal areas a little bit bigger than a grain of salt. But its temporal resolution, while good, is not the best. An EEG or EKG has better temporal resolution. So an EEG or EKG is when they hook a bunch of electrodes up to your scalp, and these little needles kind of jiggle back and forth depending on how much brain activation you have. Very good temporal resolution. EEG, EKG can capture things that are happening very fast in the brain. However, EEG and EKG have very low spatial resolution. So with an fMRI, if I pick up a difference in brain activation, it can tell you very specifically where that difference took place. With an EEG or EKG, it'll tell you with high temporal accuracy that something happened in this millisecond over here, but it's a very general over here. It's a very blurry. It happened in this general area. I can't be too specific about where. So there are trade-offs with each aspect. PET, going back to PET scans, one of the biggest disadvantages of PET scans and why they didn't take off as the main neuroimaging technology is it takes at least a minute to acquire an entire brain image. So let's say you want to know how the brain processes basic visual stimuli. Well, you would need a control condition where someone is just sitting there in the PET scanner, not doing any visual processing. So you have a baseline brain image. And then you put them in the PET scan and you have them do some visual processing. You show them some visual stimuli. Maybe you ask them, categorize this is a car versus this is a house. I show you pictures of one or the other. You're going to end up seeing some difference between the baseline brain image and the new brain image, but there's a lot of motion blur, so to speak. You're not going to get very fine-grained detail about what in particular the brain is doing at any given time because there's a lot going on in that. It takes a whole minute just to pick up on, yeah, something different is going on in here. With an fMRI, it's very well understood once the stimulus occurs and you start processing the stimulus, the blood oxygen level signal peaks pretty quickly, like within a second or two, and they make adjustments to the, when they know the stimulus onset was here at 0, and they say now correct for the actual. So they move it over and say this is when it peaked. Basically, within a few seconds, not a minute, they're able to say when a brain region changed from being relatively inactive to being more active. So with a PET scan, you would probably just see activation in the occipital lobe. That's where a lot of basic visual processing goes on at the very back of the brain. But when you're merely picking up on the fact that there's a stimulus, whenever you categorize the visual stimulus, whenever you look for abstract details of the stimulus, whether you realize the stimulus fits a pre-existing pattern, like a house versus some other thing, that's all going to get blurred out. You're not going to be able to tell if there's any specific brain region doing one specific aspect of that task. With fMRI, you will have fast enough and good enough spatial resolution to see, the very most basic aspects of the visual stimulus processing is going on at the occipital lobe, but when they categorize images, that is going on in what's called the ventral stream. There's parts of the temporal lobes that are activating. You wouldn't be able to see that with a PET scan because it's all kind of going on at the picture you get is so motion blurred that it's activation all over the place compared to baseline. And here's a great quote that really just sums up how much of a miracle boon fMRI is to people who want to understand how the brain works. In less than 10 minutes, it is possible to obtain images of the human brain that rival the quality of a post-mortem examination in a completely safe and non-invasive way. No radioactivity, you just have a bunch of magnets being magnetic waves going over you, poses no real health risk in 99.9% of people. It's really amazing. You get to just watch the human brain respond to stimuli on a very fine-grained level. So what happened with the theories underlying the brain? Well, we talked about phrenology very early in the semester. That was in the late 1800s. It probably lingered around for a few decades into the 20th century, the 1900s. But there was a big backlash against phrenology, and for a while, physiological psychologists were sort of biased against the idea that the brain has these super specialized areas that are dedicated to specific tasks. There was kind of a stigma against doing anything, doing any kind of theorizing like that. There's no specialized modules in the brain. That was phrenology, and phrenology was bad. After fMRI started picking up, it became undeniable that the brain does have some modularity. There are specific regions of the brain, and we can identify these regions down to like this specific little wrinkle here, and like the more anterior part of the wrinkle, the more ventral part of the wrinkle, which layer of cells within the wrinkle is the sixth layer. We can get very down and dirty into the what specifically the brain is doing. It's been mapped out pretty well. That's not to say that the brain is super modular. There is a lot of wiggle room in terms of how your brain is eventually going to wire itself, but it became undeniable that you can't just let the shadow of phrenology loom too large over the empirical facts. Also, lesion studies and trauma studies and neuroimaging together, kind of all contributed to this. If you take a pinprick and just kind of go to like a bird, a cat, some sort of animal, and you precisely surgically injure part of the brain, or if someone has suffered a brain trauma, such as a seizure or something, and you can, post mortem, you can look at, well, which part of the brain was gone. You can look at how damage to the brain results in damage to someone's ability to do something, such as learn categories or pay attention or plan. We used to have to actually intentionally injure animal brains with lesions or wait around for humans that had certain brain injuries. We take good notes of their case history. How is their behavior and their cognitive abilities different from normal people, and then do a postmortem. With neuroimaging, we can basically do this without all of the lesioning and trauma. You can just watch the brain in action. Neuroplasticity, like I said, the brain does have some sort of predetermined architecture in a sense. I don't believe it's ever happened that someone's primary visual area ended up anywhere other than the occipital lobe, which is at the back of your brain. The occipital lobe has to be in the cortex. It's never going to be in the cerebellum or any of the subcortical areas. So there's kind of some fixed structural specialization, but we do have this thing called neuroplasticity, where if one part of your brain is injured, a neighboring part, which will probably be doing something similar along the same lines, can take over and adaptively pick up the slack, so to speak. And parts of your brain will actually grow and change depending on what kind of experiences you undergo. For instance, there was a very famous study where London cab drivers, being a cab driver in London is very difficult. London's a very complicated city, and knowing the layout and being able to get around via car requires a lot of good spatial navigation skills and memorization and pathfinding and stuff like that. Well, one of the most important parts of your brain that does that kind of pathfinding and spatial navigation is a part called the hippocampus. If you want to remember that, you can say, I get around campus with my hippocampus. It's part of what the hippocampus does is spatial navigation, figuring your way around. Well, neuroscientists used fMRI to look at the brains of inexperienced cab drivers, non-cab drivers, and experienced cab drivers, and found that the hippocampus had larger volume in the experienced cab drivers. Now, that could be because they were doing so much spatial navigation that area received like more support and just grew out of necessity. Or it could be because people who had larger hippocampal volume happened to be good enough at cab driving to actually stick with it, and other people couldn't stick with it. The direction of causality isn't clear. All that's to say is that our brains are capable of adapting, especially the younger brain. The younger you are when you undergo some brain damage, the more likely your brain is to pick up the slack. Neighboring regions that were doing similar things will start helping out. You remember this guy, Donders? Please say you do. He always cries when people forget about him. Donders had this thing called the subtraction logic method, where you basically you want to know how long it takes someone to do a certain task. Then you have to find two versions of the task: the full task and a different version of the task that involves every part of the process except the crucial part. I should probably have that old slide here. But basically, let's say I want to know how long it takes someone to sort a stimulus as either a red light or a green light. Okay, that's the full task. How long does it take them to do that? Well, you're going to have them do everything but that. So when you emit a behavior indicating that you have categorized the light as red or green, first you have to see that there's a stimulus. Then you have to categorize it. Then you have to initiate the motor response to press the button to show that you know if it's a red or a green light. There's a long process, and only part of it is actually categorizing the stimulus. So what Donders said is if you want to measure just the part of interest, you have to have the full task minus everything but the crucial task, and then you'll have the leftover reaction time. That's the part you really want to know about. Well, fMRI in the olden days, though these days are past, but fMRI basically used the exact same method. They would say, you want to know which part of the brain categorizes stimuli? Well, you have to have someone do the full task. I show you stimuli. You perceive that there's something there. You pick up all the low-level visual details. You pick up on higher-level details. You categorize it as the pattern, overall pattern belongs to one category or the other, and then you execute the motor response. I'm going to press this button because it belongs in this category. You have someone do the full task, and then you have someone do everything but that last part. So that might mean I'm going to show you a stimulus, and regardless of which category it belongs in, you just press a button as soon as you see it. So all the things I just said, something is visible now, so we're activating one part of the brain. Now we're starting to pick up low-level details, so that's different parts of the brain that are activating. Starting to pick up on higher-level details. We're building up the stimulus visually. Then, okay, you've seen it. All you have to do is press the button because you've seen it. So we're going to initiate the motor signal, and we're going to press the button. Well, if you have the brain activation data from those two tasks, you can just say, take all the brain activation that was going on from the full task and subtract out the parts that were going on with everything but the main thing. So take out the motor movement, take out the basic visual processing. What you're going to end up with is the full task is going to show activation in the occipital lobe, which does low-level visual processing. It's going to show activation in the ventral stream, so going down to the ventral part of the temporal lobes. That's part of the actual pattern recognition. And then the motor strip on the top of your head is going to be involved in initiating the motor response, pressing button. You have the full task there. You subtract out the part on trials or conditions where you were just pressing a button whenever a stimulus shows up. So take out the occipital lobe, take out the motor strip. You're left with just the ventral part, the temporal lobes. So you've pinpointed where a specific part of the task goes down. So Donders really got his appreciation after all. Everyone used his method, at least for a while. Whenever you acquire an MRI image, you're actually acquiring a series of voxel values. A voxel is a three-dimensional pixel. Your phone has a, so many thousands of pixels by so many thousands of pixels two-dimensional display, meaning it just has a height and a width. There's no depth. You don't have pixels under your pixels on your phone screen. You don't have any pixels above the pixels of your screen. It's a two-dimensional display. For a MRI analysis, you have voxels, which is just, I've got one layer height and width. I can tell you where that frontmost layer, I can locate any block in that layer. But then there's a layer underneath that one, and that has a different set of blocks or voxels. So what an fMRI does is it's basically got this big grid. It's got an x-axis, a y-axis, and a z-axis for depth. And it places each part of your brain into one of these voxels and tells you how active the brain is in that voxel. Fun fact, all of the programming underlying the Minecraft video game uses the same voxels and whatnot. Each voxel represents a 1 to 3 millimeter on each side, height, width, depth. Pretty small. It's I think a little bit bigger than the width of a human hair if you cut it. There's something on the order of magnitude of 100,000 voxels. So these voxels are actually way too big because there's not 100,000 of those. This is just to help you visualize it a little bit. And that's what we're dealing with when we do fMRI data, is we're just saying, look, here are the voxels for when the brain is doing this. Here are the voxels for when the brain is doing that. You subtract these activation voxels from these, and you're left only with the activations associated with the part you want. That's at least the old school way of doing fMRI data analysis. There's a newer technique called multivoxel pattern analysis, or MVPA, which kind of just amounts to a kind of weird mind reading. I'm not going to go into too much detail about it because I feel like I've already gone into way more detail than strictly necessary on the old school fMRI data analysis, but I did want to just whet your appetite a little bit for it. There's also something called model-based fMRI, where instead of comparing voxel to voxel or region to region or doing MVPA, you're creating a model saying at this point in the task with these stimuli, the brain, you have a model saying what the brain should be doing based on this theory. It's pretty complicated, but this is kind of the cutting edge of how people do neuroimaging studies. So I talk the most about fMRI because that is by far the most popular neuroimaging technique. It is not the only one. I talked a little bit about EEG. That's when you have a bunch of little electrodes hooked up to your cap. You got a little skull cap, and you have readings that are very fine-grained temporally. It gives you lots of moment-to-moment information, readout like that. Very fast neural activity recordings, but poor spatial resolution. So if you were having, this is where the prefrontal cortex is on this guy. If he's doing something that involves the prefrontal cortex, this electrode will pick up that it's happening somewhere in this general vicinity, whereas an fMRI will be able to tell you, no, like zoom in, zoom in, zoom in. It's going on right specifically right here in this little teeny tiny fold. fMRI has much better spatial resolution than an EEG or EKG, but EEG and EKG have better temporal resolution. So depending on what you're studying, it might be more important to have temporal resolution. If these mental processes are going on quickly, then an fMRI is not going to cut it. You also have something called TMS, transcranial magnetic stimulation. It's basically a magnetic wand that will overstimulate certain brain regions underneath the wand, stimulate them so much that they'll become inactive. This is safe. We wouldn't just do things to people that isn't safe. So what you can do is you can run a study and say, what happens if someone's, whatever, this little bump right here, if we disrupt the function in that little lump in the brain right there, are they still able to comprehend sentences, or can they still can they still produce sentences? You can get information of what brain regions are doing what based on which ones need to be working in order for you to do certain things. It doesn't have the best spatial resolution, though. I said it, disrupts this one little fold, but actually it disrupts a fairly large area of the brain, like the brain mass underneath it, and only directly underneath it. So it can only affect the outermost layer of the brain, which is called the neocortex. A lot of important stuff is going on in subcortical regions deep into the brain, and a TMS wand cannot really do anything to those. So there are limitations to TMS. There are also electrodes that are implanted directly into the brain, or electrode arrays where you have multiple electrodes. This is not usually done it's never done just for scientific purposes. People will get electrodes implanted in their brain for medical reasons, because something needs to be monitored, or because it's a treatment to be able to remote control, like, I want these electrodes to start going off. There are legitimate medical reasons for why these would be in your brain. You wouldn't just put them in there because some scientists want to know what this bump does or this bump does. Electrodes have the best spatial resolution because in many cases they're just recording the activity of one single neuron. You really can't get any better than that. Now, whether it's useful to know what one single neuron is doing is a matter of debate. I personally think it's not very useful, but I'm biased. I'm an fMRI guy. And then, of course, we have old-fashioned lesion studies, which thankfully is not terribly necessary anymore. And it also has drawbacks, because if you have a lesion and you're still trying to function and rehabilitate yourself, then that kind of muddies the waters, trying to infer what is this region usually used for. If you've lost the ability to do certain things mentally, but you're also undergoing rehab, so neuroplasticity, your other neighboring regions are taking over. It's kind of muddy waters trying to figure that out. And lesions can be very big and sometimes very messy to try and there are the mere fact that you have something going, I don't know, it's not the cleanest neuroimaging technique. It's not even a technique or technology. It's just an unfortunate circumstance. All right, we're almost done, I swear, with part 2, and then we're going to go to part 3. So, neuroimaging in the last quarter of the 20th century has a strong effect on science, on psychological science. For one, if you are going to be a major figure in psychology and you're going to contribute to our theories and understanding of psychology, you have to line up with, your theory has to line up with the brain. It has to be neurologically plausible. The age of Freud, where you can make up these theories that vaguely resemble some stuff your patients did, are long gone. You have to be pretty firmly connected to empirical observations of the brain. Everything has to be neuro-based. And the money. It costs at least $40,000 to do a modestly effective fMRI study. It costs about $500 an hour to reserve an MRI and use it. So what that means is, if you want to do any fMRI research, your university has to have one. First of all, it has to be a pretty well-funded university, and universities don't tend to spend millions of dollars not only to acquire an MRI, but then to maintain it and have staff to run it. And if you need to repair it, it's almost cheaper it's not cheaper to buy a new one, but it's very expensive to repair or replace parts for an MRI. And, you need people that know how to run it. MRI can't just be put in a broom closet. It has to be in a special facilities that meet certain conditions, because you have the high-powered magnets. Universities don't tend to invest that kind of money in an MRI for it to just sit there and just, not get used and not produce its own income. They want you as a researcher to pull in your own funding. So, psychological science has kind of become a game of who can get their MRI study funded. It's a very expensive, high stakes game. You need to get the NSF, that's the National Science Foundation, or the NIH, and that's National Institute of Health. Private institutions like charitable organizations might support your research. Uh, Scientists have kind of become fundraisers. A big part of their job is raising funds, and stakes are high. If you put your neck out there and you worked hard to compete and get yourself the $250,000 to run this study, that's a lot of pressure. I hope your study really works out, because if it doesn't, that's a lot of money you just lost. But we'll get more into that in Module 7. But yeah, money plays a big role in psychology now because of all this expensive neuroimaging technology. And I'm going to actually spare you this talk about backwards inference. I think it is important, but I have to pick and choose what I'm going to cover. I covered this last time. I still think it is important, but I don't know. I covered the really important stuff, so I'm going to skip this. If you are interested at all in neuroimaging techniques, I cannot recommend this book highly enough. It's called The New Mind Readers. It's written by Russell Poldrack. Poldrack is the guy for MRI, fMRI data analysis methodologies. And this book is very accessible, probably more accessible than me. It goes over the history and the benefits of these different technologies, and he talks about some of the latest techniques used to read minds, basically, and goes over multivoxel pattern analysis, which I kind of just said, yeah, I quit. I'm not going to try and explain that in passing. Very good book, very accessible. Eliminative materialism is another concept. Backwards inferences are bad, and Russell Poldrack does explain that. I'm not going to test you on that, but I will quickly say eliminative materialism is not an idea that is taken very seriously by a lot of people, but it is a good thought experiment to run yourself through. Basically, eliminative materialism is the view that there is no mind, there's just a brain. Humans are these brains that are piloting bodies. And this common sense stuff we use in our folk psychology, that's what they call our ordinary concepts that we use to describe behavior, including our own mental processes, other people's mental processes. That's folk psychology. Talk about beliefs, emotions, strategies, concepts, etc. According to eliminative materialism, eventually these concepts are going to be eliminated. It's going to turn out that talk of beliefs and emotions and sadness are going to end up being these crude concepts that had no empirical basis whatsoever. Kind of like how spirits and witches and curses used to be considered a real thing until we learned better. Eliminative materialists say we're going to learn so much about neuroscience that we're going to have this revolution that all of our old concepts are going to go out the window, and we're just going to talk with neuroscience-based vocabulary about people's thoughts and behaviors. That's about as deep as I want to go into that. I think you should be aware of eliminative materialism, especially if you're going to appreciate contemporary psychological science, which is so heavily based on materialist views of the mind and the brain. But that is everything for the brain. I'm going to finish. Actually, no, there's more. Part 3, there's going to be a part 4 too. But this is the end of part 2. So take a break and come back later, because I certainly am. And I will see you for part 3.